Hello everyone, myself Dr. Tejas Shah. I am working as an Associate Professor, Department of Biochemistry at Srimati B.K. Shah Medical Institute and Research Center, Sumandi Vidyapeeth, Vadodara, Gujarat. The purpose of this channel is purely a teaching and learning. Today we will see the metabolism of protein, digestion and absorption. The session is drafted for first MBBS students. The learning objective for this session: at the end of this session, the learner will be able to describe the digestion of proteins, explain the absorption of amino acids, explain Gamma-Glutamyl Cycle, also called Meister Cycle, describe intracellular degradation of proteins. First, we will see the metabolic interrelationships of amino acids. Proteins are made up of the polymers of amino acids linked together by a peptide bonds. So the sources of the protein could be the dietary proteins is provided in the diet, and secondly the proteins which are not essential, means they are synthesized by our body. So you can see here the dietary protein undergo the digestion to provide amino acids. These amino acids go to form apart various important body proteins. These body proteins are undergo also the turnover, the degradation. So this is a daily turnover of the protein. The amino acids further broken down into the two parts. One contains the nitrogen containing part, another is the nitrogen free intermediates, also called a carbon skeleton. It can be used for the production of the glucose, ketone bodies, and used for the energy purpose. They also used for biosynthetic products also. The amino part, that is ammonia, enters into the liver to form the urea further excreted by the kidney. So if we look the overview of the protein metabolism, so protein have two important sources. One, it is the intracellular proteins, and secondly which is provided in the diet. So these two process we are going to discuss today, that is digestion and absorption, that will lead to the formation of the free amino acid in the blood. Then this amino acid is further catabolized to form the nitrogen part, and second is the carbon skeleton part. Both these pathways are different, but they are interlinked together. So we will discuss today the digestion and absorption of amino acids. So let us begin with the proteolytic enzymes. We all know that cooking helps in the digestion. The cooking causes the denaturation of proteins, means disruption of the quaternary, tertiary and secondary structure that open up the site for the enzymatic action, so that the digestion can take place easily. All the proteolytic enzyme belongs to the class 3 hydrolases. They are secreted as an inactive zymogens and active from in the intestinal lumen. So proteolytic enzyme broadly classified into two: endopeptidases and exopeptidases. Let us see. As you can see here, the two part, endopeptidases and exopeptidases. Endopeptidases, you can see here in this diagram the arrow mentioning the action of this endopeptidase. These endopeptidase, they act on peptide bonds inside the protein structure or the protein molecule, so that protein becomes successively smaller and smaller units. And this group of endopeptidase contains the pepsin, trypsin, chymotrypsin and elastase. Exopeptidase, which act at the peptide bond only at the ends of the polypeptide chain or the protein molecule. They are further of two types: carboxypeptidase and aminopeptidase. Carboxypeptidase means they acts on the peptide bond only at the carboxy terminal end of the chain. Here you can see the carboxypeptidase. So this is the carboxyl terminal end. So they act here. And aminopeptidase, you can see that is an amino terminal end, so that they act here. Okay, and further the tripeptidase and the dipeptidase belong to the exopeptidases types. So digestion of proteins, if you compare with the carbohydrate digestion starts right from the mouth, but the digestion of proteins do not occurs in the mouth, it will start in the stomach with the help of the HCl, hydrochloric acid and pepsin. Pancreas with the help of trypsin, chymotrypsin, elastase and carboxypeptidase, and continue in the intestine with the help of aminopeptidase, dipeptidases and tripeptidases. So if you look in this diagram, the importance of the gastric glands in the stomach lining. So the parietal cells which provide the important hydrochloric acids, and second the chief cells that will secrete the pepsinogen, and this secretes under the effect of the hormone gastrin. Second important aspect is the exocrine cells of the pancreas that will release the pancreatic juice which contains the bicarbonate and the pancreatic enzymes. And third is the villi of the small intestine that will provide the site for the absorption. Let us see first the gastric digestion. It is initiated in the stomach. It's an important hormone that is renin. It is also called chymosin. It is different from renin. The renin is the proteolytic enzyme that is in the kidney tubules. It converts the angiotensinogen into angiotensin, a hypertensive agent. But this renin is active in infants and involved in the curdling of the milk. It is absent in the adults. So the infant mainly depending on the milk. So milk protein, especially the casein, is converted to the paracasein by the action of the renin, and further this paracasein is broken down with the help of the pepsin. Hydrochloric acid secreted from the parietal cells which provide the optimum pH for the enzymatic action, that is 1.6 to 3.2. It denatures the quaternary, tertiary and secondary structures and activates the pepsinogen to form the pepsin. The pepsinogen is secreted from the chief cells. So the activated pepsin cleaves the bond formed by the carboxyl group of the phenylalanine, tyrosine, tryptophan and methionine. So when the digested product leaves the stomach, it is the mixture of insoluble proteins, soluble proteins, peptides and amino acids. So if we see in this animation, so upon the activation pepsinogen with the help of the hydrochloric acid, it is converted to the pepsin, as it is an endopeptidase, so acts on the in between the peptide bond like this. So continuation digestion in the pancreas. So when the acidic chyme enters into the duodenum or the small intestine, the hormone secretin is released, so that the pancreas release the bicarbonate, which helps to provide the optimum pH along with the bile. And secondly, under the effect of cholecystokinin and pancreozymin, the pancreatic enzymes are released. Now activation of the pancreatic enzymes. So these are the pancreatic enzymes: trypsinogen, chymotrypsinogen, proelastase and procarboxypeptidase. Trypsinogen is activated with the help of enterokinase. This enterokinase is also called enteropeptidase. So it converts the trypsinogen into the active trypsin, and this trypsin then further activates other these proenzymes to form active chymotrypsin, active elastase and carboxypeptidase. The important clinical aspect to note down here: the premature activation of the trypsin will destroy the pancreatic acinar cells and lead to a life-threatening condition called the acute pancreatitis. Now we see the action of the activated enzymes. Trypsin break the bond of the break the bond which are formed by the carboxyl group of arginine and lysine. Chymotrypsin break the bond formed by the carboxyl group of aromatic amino acid and methionine. Elastase will break the bond formed by the carboxyl group of alanine, glycine and serine. Carboxypeptidase A will break the bond formed by the carboxyl group of branch chain amino acid and alanine, and carboxypeptidase B break the arginine and lysine. Now, intestinal digestion. Complete digestion of the small peptides to the level of amino acid is brought about by the enzymes present in the intestinal juice, that is called succus entericus. The luminal surface of the intestinal epithelial cell contains the following enzymes: leucine aminopeptidases, which cleaves at the amino terminal of basic amino acids and glycine; proline aminopeptidase, which cleaves the proline from the ends of the peptides; dipeptidases and tripeptidases, which cleave the dipeptides and tripeptides respectively. Now the absorption. So absorption of amino acid occurs mainly at the small intestine, where we receive the digestive products of the protein, mainly the free amino acids, tripeptides and dipeptides. So we see the absorption of each of them. First, the free amino acid absorption, that is the sodium dependent carrier transport system. These are the carrier systems for neutral amino acids, basic amino acid, acidic and amino acids. These carrier systems requires energy, that is called a secondary active transport. Let us see in this diagram how it is occurs. So amino acid along with the sodium enters into the cell. So this is called a symport. Once the sodium enters into the cell, we know that the sodium is most importantly or most abundant cation extracellularly. So to shift this sodium, the important transport system is the sodium potassium ATPase pump, which throws sodium outside and potassium enters into the cell, and this requires the energy. Whatever amino acid enters into this cell is further transported into the portal way by the facilitated diffusion. Okay, so this is called a secondary active transport. You can compare it with the sodium dependent glucose transport that is occurring in the carbohydrate metabolism. Now the peptide absorption. So peptides, dipeptides and tripeptides, so their transport is somewhat more rapid than the free amino acids, and that too occurred with the help of this carrier, peptide carrier. It is also requires the energy. So once the peptide enters into the enterocyte, it is further broken down into the free amino acids, because only the free amino acid absorb into the blood circulation. Absorption of the intake proteins. It is mainly important in case of the newborns, where the first 24 hours after birth, immunoglobulins get absorbed and it provides the passive immunity to the newborn. But in adults, it has a very little nutritional significance. That is, when an intake protein is absorbed mainly by the paracellular routes, that is tight junctions provided between the cells, and by the intracellular routes, that is endocytosis and pinocytosis. But these larger molecules, when they pass, sometimes they act as a immunogenic and causes antibody reaction leading to the food allergy. The allergy to certain food proteins, that is milk and fish, is believed to result from absorption of partially digested proteins. Now the important aspect regarding absorption, that is gamma-glutamyl cycle, also called Meister cycle. This is used for absorption of neutral amino acid in intestine, kidney, tubules kidney tubules and brain. So the main role is played by the tripeptide called the glutathione. It is also called gamma-glutamyl cysteine glycine. So it reacts with the amino acid to form gamma-glutamyl amino acid. You can see the amino acids from this enters into and combined with the glutathione to form gamma-glutamyl amino acid, and cysteine glycine is liberated, further degraded to form glycine and cysteine. This gamma-glutamyl amino acid is cleaved to release the free amino acid here inside the cell, and there is formation of 5-oxoproline, and the enzyme is 5-oxoprolinase. This 5-oxoproline is converted to the glutamate with the help of ATP. Further, it is received the cysteine and combined to form gamma-glutamyl cysteine with the help of ATP, and further it receives glycine to further regenerate the glutathione, and this glutathione again receives another amino acid and help its absorption. So here you can see there is 3 ATPs are used in this entire cycle. Now if you look the clinical aspect of this cycle, if there is a blockade here, means absence of this enzyme 5-oxoprolinase, it will lead to the condition called oxoprolinuria. Other diseases of the absorption are also notified, that are Hartnup disease and cystinuria. We will discuss in detail when we discuss the aromatic acid metabolism as well as sulfur containing amino acid metabolism. There we will discuss this in detail. But remember at present that these are the diseases which are related to the absorption of the amino acids. So if we see the summary, you can see the digestion start at the stomach. Proteins are degraded with the help of HCl and pepsin to form small peptides, large peptides and free amino acids. In the intestine, the pancreatic proteases also act. Aminopeptidases, dipeptidases will further broken down this into di and tripeptides. So at the level of the brush border, they are enter with the help of the carrier into the enterocyte. They are absorbed and further they are degraded to form free amino acid that can be transported into the blood. Here free amino acids are also transported to the carrier mediated transport, which is mainly the sodium dependent that we have seen the various carrier systems, and thus the amino acids are absorbed. Now we see the important aspects of the intracellular protein degradation. So the rate of protein synthesis is just sufficient to replace the protein that is degraded. So we have seen previously that there is constant turnover occurs with regard to the proteins. So the half-life of short-lived protein, for example, the proteins which are mainly the regulatory proteins and the misfolded proteins, they are short-lived protein, so their half-life is found that is in minutes or hours. Half-life of long-lived proteins, for example, mainly the proteins found in the cells, their half-life is days to weeks. And half-life of structural proteins, for example collagen, found to be months and in years. So two important enzyme systems which are responsible for intracellular protein degradation are ATP-independent degradative enzyme system of lysosomes. Second is the ATP-dependent ubiquitin-proteasome system of cytosol. But before that, what are the signals that will lead to the protein degradation? So chemical signal for protein degradation. So protein degradation cannot be a random process, because as we have seen, the proteins have a different half-lives, and so it is also influenced by some structural aspects of the proteins. Whether there is structurally change occurred in the protein or not, that will also affect its degradation. Here we take the example that protein that have been chemically altered by oxidation, or either they are tagged with the ubiquitin, are preferentially degraded first. The half-life of a protein is influenced by nature of the N-terminal residue also. Recently, it is identified that proteins rich in sequences containing the proline, glutamate, serine, and threonine, collectively called as a PASTE sequences. This name PASTE is derived after the one letter designation of these all four amino acids. So, the proteins rich in the PASTE sequences are rapidly degraded and therefore they have short half-lives. So ultimately the chemical sequences specifically tagged or having the specific sequences are selectively or preferentially degraded. Now let us see the first system, ATP independent degradative enzyme system of the lysosomes. You all know the lysosomes are the bags of enzymes. So they use the acid hydrolysis and the protein called cathepsins. Cathepsin is the Greek word, its meaning is to digest. So two important mechanisms by which lysosomes carry out the degradation of protein. One is the degradation of the intracellular proteins, that is called autophagy. Here one example, if you see in the diagram, the mitochondria will form the autophagosome. This autophagosome is combined with the lysosome to form autophagosome, and the action of the lysosomal enzyme will form the phagolysosomes, and further the protein degradation products are released. Secondly, it degrades the extracellular proteins, that is called heterophagy. So these two mechanisms are there in the lysosome for protein degradation. Now second important system is ATP dependent ubiquitin-proteasome system of the cytosol. So there is a constant turnover of protein. Intracellular protein breakdown also occurs independent of the lysosome, that is occurs in the cytosol with the help of this ubiquitin-proteasome system. So this involves the important ubiquitin protein. It is a non-enzymatic protein. It is so named because it is seen in all the cells abundantly. It is having 76 residues in its structure. So the proteins marked for the degradation by covalent attachment, this is the protein which is need to be degraded. It is tagged by this ubiquitin with the help of the three important enzymatic action. Okay. So ubiquitination of the target substrate or the target protein occurs through the isopeptide linkage of alpha carboxyl group of C-terminal glycine of ubiquitin, C-terminal glycine of ubiquitin to the epsilon group of a lysine on the protein substrate by the three-step enzymatic reactions. How this enzyme takes place, reaction takes place. So enzyme 1, it is called the activating enzyme which activates this enzyme E1, it is called activating enzymes that activates the ubiquitin, which is then transferred to the next enzyme that is called E2, a called conjugating enzyme. And third, the enzyme E3, it is called the ligase enzyme that identifies the protein to be degraded and interacts with the enzyme 2 ubiquitin complex. So the consecutive addition of the, you can see here, consecutive addition of these 4 or 5 ubiquitin molecule here, that will helps the catalytic protein, it is called 26S proteasome system to identify it. So this 26AS proteasome, if you look here into the structure, it have two portions, that is 20S core and 90S regulatory particles. These 90S regulatory particles are also called caps, and this 20S core mainly contains the catalytic activity. And here you can see this is the targeted or protein needs to be degraded. Along with this is the ubiquitin molecule. So polyubiquitin attached to the substrate protein which interact with the proteasome. So 26AS proteasome identified this protein to be degraded, tagged with the ubiquitin. So 90S, that is cap protein or the 90S regulatory particle, will remove the ubiquitin, and it directs the path of this protein to be degraded inside the 20S proteasome core. And inside this core, the degradation takes place, and these protein particles, amino acid particles, are liberated. So they liberated as amino acids, or they can act as antigen presentations. So unlike the system which is operated in the lysosome, here the ATP is used. So in nutshell, we have discussed the amino acid pool mainly provided by the dietary protein through the digestion and absorption process, and secondly the body protein broken down with the help of two important systems. One is operated in the lysosome and another is operated in the cytosol, called the ubiquitin-proteasome pathway. These are the references based on which the presentation is made. So thank you everyone for your kind attention and listening. So like, share and subscribe this channel and click the ring bell button to get the notifications. Thank you everyone.